Our next speaker is Professor Koen Venken, who has a long-standing interest in T-cell biology with the primary focus on translational research. He completed his PhD at the Biomedical Research Institute of the University of Hasselt in Belgium and Maastricht in the Netherlands, looking into the role of human Tregs in multiple sclerosis. For his postdoctoral training, he moved to the University of Ghent in Belgium, becoming acquainted with human and experimental research in the field of rheumatology. And for this, he received grant support from the Research Foundation Flanders in Vlao. Dr. Venken is currently a senior staff scientist at the Flemish Institute for Biotechnology, Center for Inflammation Research, and appointed as an assistant professor at the Faculty of Medicine and Health Sciences of Ghent University. And his current research focuses on the role of innate-like and regulatory T cells in the area of chronic inflammatory diseases, specifically joint and mucosal tissue inflammation. And he'll be talking to us today about understanding the role of T cells in combined gut and joint inflammation.
Good afternoon, everyone, and thank you for the kind introduction. I hope you can see my slides on full screen now. Hello?
Yes. Yep.
Perfect. So I will give a presentation about our recent publications on Tregs in a TNF-driven spondyloarthritis model. And I will first start with a short introduction. So spondyloarthritis is actually a cluster of rheumatic diseases which show and share genetic and physiopathological features. So these patients, they can present with which we call axial or peripheral inflammation. And so the axial inflammation can be present in the sacroiliac joints, or even in the spine. Prototypical disease you're probably familiar with is ankylosing spondylitis, and but it can also present with peripheral manifestations with, for instance, a knee synovitis or an Achilles tendon enthesitis. But next to these joint manifestations, let me close that. Yeah, these patients also show extra-articular manifestations like skin psoriatic-like disease. And also what we discovered quite a while ago is that almost 50% of the patients show microscopic signs of gut inflammation. And we have a keen interest in this so-called gut-joint axis in spondyloarthropathy. So why is this important? So it's important because it's a major risk factor to develop actual Crohn's disease. So 5 or 10% of the SPA patients will develop Crohn's disease. And patients who already show chronic signs of microscopic gut inflammation, even 1 in 5 of the patients will develop Crohn's disease. So it's a major risk factor for IBD. And on the other hand, it also leads to a higher risk of structural joint damage. As seen here on MRI of the pelvis, there is an increased bone marrow edema present in patients which have these chronic or acute signs of gut inflammation. So the gut-joint axis is a very topical field of investigation because it also have medical reasons to look into this, because of course some therapies which are effective in IBD are not in rheumatic disease and vice versa. So it's always the question we have is this, what's first there? Is there first gut inflammation and then joint damage, or vice versa? So we know that IBD patients can also develop rheumatic diseases. So that's still a topic which is heavily investigated and we're looking into as well. So there are many factors. So it's a multifactorial disease. So many factors are involved in this kind of chronic inflammatory diseases. We know the genetics are important, and of course also the environmental factors. And yeah, nowadays you can't go around the microbiome anymore. So also microbial factors can be important. And we think this interaction can lead to some chronic stage of immune dysregulation as pro-inflammatory cytokines like IL-23 and IL-17, but also TNF-alpha are very important in the disease pathogenesis, as shown by the effectiveness of anti-IL-23, 17, and TNF-alpha biologicals. Although there is still an unmet medical need because quite a fraction of these patients, up to 50%, do not respond to these kind of biologicals. So we need to know more about the pathophysiology behind these disease manifestations. And so in this study, we used a mouse model called the TNF-delta ARE mice. So these mice, which were initially developed by George Collier's team in Greece, these have a deletion of AU-rich regulatory elements in the TNF gene, by which they will express higher levels of TNF upon triggering. And this will lead to both joint pathology and gut pathology in these mice. And this resembles the human disease. So it's, of course, every model is a model, but this is quite a nice model to study the combined gut and joint inflammation as seen in patients. And so the first thing we did to have a better insight in the effector and regulatory pathways which are involved in upon TNF overexpression in the gut and the joint is actually to do an unsupervised, unbiased transcriptomic analysis. And for this, we isolated ileum samples and the synovium samples from the mice, both from the TNF ARE and the wild-type mice, and we did bulk RNA-seq. And you can see here from the volcano plots, there were quite some genes overexpressed both in the inflamed ileum and the synovium. So you actually get a very rich data set, which get you kick-started on investigating some of these interesting pathways, of which we potentially have tools in-house. So in all of these genes, you will look at in the families of genes to have an idea what is going on. And so we looked in certain cytokine genes, TNF family related genes, and cell marker genes. And as expected for the cytokine genes, we saw actually the confirmation that TNF was upregulated. But you can also appreciate from this figure that IL-1 beta and IL-33 are also upregulated, both in the gut tissues of the inflamed, of the pathogenic animals, as well as in the joint samples. But there are also some site-specific upregulations going on. For instance, IL-6 can only be detected in the synovium, whereas, for instance, IL-17A is mostly expressed in the inflamed gut samples. So that's quite intriguing that secondary inflammatory loops are ongoing and site-specific. The same was the case for these TNF-related family members. And I will not go into detail now on this figure, but it comes back in later in our presentation. We saw also here some site-specific changes. So that's quite intriguing. And for the cell markers, of course, given this is an inflammatory disease, you will have increase of myeloid cell markers, mostly from macrophages. But also what we found intriguing was the induction of FOXP3, which is a key master transcription factor for Tregs. So first we were intrigued to what happens with TNF and its receptors. So TNF signals via two receptors, TNF receptor 1, which is pleiotropically expressed, and TNF receptor 2, which is more restrictedly expressed on certain cell types. And if we looked at the gene expression of these two TNF receptors, we saw actually that regarding the TNF receptor 1, not a lot of changes were detected. So there was an increase in the synovium as compared to the ileum, but there wasn't really an induction upon inflammation. At least it was not significant in our setup. But what we did see was that the receptor type 2 was clearly induced both in the synovium as well as in the ileum. And so we were intrigued by this selective TNF receptor upregulation. So we also looked at the protein levels. Normally the receptor are, of course, expressed at the cell surface, and that's not depicted here. We could not see any difference in the protein expression between cell types, lymphoid or non-lymphoid, in gut versus joint. But what we do know is that these receptors are also cleaved. That's an ongoing procedure and especially accelerated upon inflammation. And if we looked at the soluble TNF receptor 2 levels in the serum of these mice, we could indeed see a progressive increase of this soluble TNF receptor 2. And this coincided with the induction of an enzyme called ADAM17, which encodes TACE, so TNF alpha converting enzyme, which is responsible actually for the cleavage of not only TNF, but also the TNF receptors. So that's coincided nicely. So has this any human relevance? So to address this question, we also take the opportunity to look in some serum samples of patients. So we had a large collection of SPA serum samples, and we compared it to other rheumatic disease, excuse me, called rheumatoid arthritis, but also patients with inflammatory bowel disease next to controls. And whereas for the soluble TNF receptor 1, there was no significant difference between these patient groups, we saw clear increase or the highest levels of soluble TNF receptor 2 in SPA patients as compared to the other groups. And quite strikingly, if we then would stratify the patients, whether or not they have inflammatory gut signatures, which we investigate upon intakes of these patients, because in our clinical cohort, the patients undergo an ileocolonoscopy at baseline. So even before treatment, we have an idea about the processes starting to go on in the gut. And here you can see that the patients who show gut inflammation on histology also have the highest expression. So bringing this story together, that the gut inflammation is really important in this. So as I mentioned, TNF receptor 2 is only expressed by a limited number of cell types and is known to be highly expressed by Tregs. I don't think these cells need any introduction to this audience, but Tregs are, of course, a master suppressor cell. As you can appreciate from these plots, you can see there was an increase in FOXP3 CD25 positive CD4 T cells. And this was both in the gut tissue as well as in the joint tissues apparent. And these is the quantification of that. So these were significantly increased numbers of Tregs. So the Tregs seem to be attracted to or locally amplify in the inflamed tissues. And what about their TNF receptor 2 expression? So we expected high expression, and that was indeed the case if you looked at the lymph nodes. So the draining lymph nodes of the gut and the joint tissues showed a high TNF receptor 2 expression, whereas TNF receptor 1 was not expressed. But if we then looked in the tissues, also in the ileum samples, we saw that the TNF receptor 2 was highly expressed. But in contrast, in the synovium samples, so of the ankle, there was really a significantly diminished expression of TNF receptor 2, which again, you see the quantification here. And we compared it also to conventional T cells, which almost have no expression of this receptor, TNF receptor type 2. And upon inflammation in the ileum samples, you could also see a decrease already of this expression. So probably mirroring the effect we saw with the soluble TNF receptor release. And consistent with the TNF receptor mRNA turnover. So how do these Tregs behave and upon high TNF load? So TNF, we know there is some conflicting literature. Some people say Tregs, or have shown that Tregs are inhibited by high levels of TNF. Others see the opposite. They actually strengthen their function. So we tested in our own system. In the mouse system. So we did it first in vitro. So we knew the lymph node Tregs expressed high levels of TNF receptor 2. We took out these cells and conventional cells and we put them in a classical co-culture assay. So we have unstimulated conventional T cells. We label them with a cell proliferation dye, like CFSE, reduced eFluor 670. And then you can, after polyclonal activation, you can measure the actual proliferation of these cells. And if you add now Tregs, you can see that both for the TNF ARE as well as the wild-type mice, these cells were really able to suppress the proliferation of conventional T cells. So this seems to respond normal. If you looked into the cytokine production by the conventional T cells, it gets a little bit more complicated. So both the TNF ARE here in red and the white representing the wild-type Tregs were able to suppress the TNF and interferon gamma secretion, although this was clearly diminished in the TNF ARE mice. But in extension to that, we did some crossover experiments, and this is quite an elegant way to see which factor is predominating this apparent disability of the Tregs. So for this, we took conventional T cells from the TNF ARE mice and cold cultured them either with TNF ARE Tregs or wild-type Tregs, and we also did the opposite. So you can see here that TNF ARE and wild-type Tregs are almost in the same range of suppressing the conventional T cells. So we see that there is actually some restriction towards the ability of Tregs to suppress this TNF response, as the opposite experiments, so wild-type conventional T cells with Tregs from both the different mice, show that it was actually easy for them to suppress the TNF. So there is some restriction of conventional T cells, whereas the Tregs seem to behave normal under these high TNF exposure conditions. So what happened in vivo? So we want to know, does the impact of the high TNF have a functional impact locally in the joint on the gut? And for this, we use the Treg mice. So Treg are depletion of regulatory T cell mice, which have a bacterial artificial chromosome, including the diphtherin toxin receptor. And by means of adding DT to the mice, you can actually elegantly deplete the cells, the Treg cells in this case, in your mice. And it also has an EGFP tag, so you can also use these mice as a reporter, which is quite handy if you want to sort the Tregs. And in comparison, we used NKT cell deficient mice, which we know are profound suppressors of both joint and gut inflammation in this model. So what we saw is that looking upon the joint inflammation by visual scoring, that there was actually no difference between Tregs and TNF ARE non-Treg mice, whereas you could see there was still a window for disease progression at the G-alpha 18 knockout mice clearly had a more progressive disease. And these data were consistent with the histological assessment we did, and we also did some functional testing. Sorry, I have some difficulties with this banner. But we did a grip test where you can put the animals on a grid and measure the actual power they have in their front and hind paws. In contrast, if we looked at the gut inflammation, we saw also in the Treg mice quite severe amplified phenotype. So there was a stronger inflammatory infiltrate, and we observed clearly these more blunted villi in the Treg mice versus the TNF ARE mice. So in summary, it means that although both the Tregs in the synovium and in the gut are exposed to high levels of TNF, the cells in the joint are not capable of suppressing the ongoing inflammation, where in the ileum it's clearly going on. So to understand the differences between these two Treg subtypes, we did a bulk RNA assay where we sorted the Tregs from the synovium and the ileum, and also of the draining lymph nodes. So this was quite an intensive experiment. So you have to be careful that you sort the right cells, enough numbers, and then do a careful RNA prep, etc. And it's intriguing to see if you then do a principal component analysis of this transcriptional data set. You could see that the cells coming from the lymph nodes were clearly separated from the ones coming from the gut or the joint, as shown here by the first component. But also the gut and joint Tregs were quite different regarding their transcriptome profile. And we could also see already here some differences in the TNF ARE versus the wild-type mice. And this was illustrated here as well in this heat map showing the clustering of the different Tregs, primarily based on the origin of the cells coming from lymph nodes or the tissue, and secondary to the inflammation. So if we looked at the differentially expressed genes, we could see there were quite some in the synovium and ileum, and there was not a lot of overlap with the draining lymph nodes. So meaning that it's really a local imprinting of the Treg cells going on, whereas it seems not to be a recruitment of these cells from the lymph nodes which already been exposed to high TNF in these lymph nodes. So this was quite intriguing. And then we looked back, so we had a lot of data to look into. What was intriguing to look at is again these TNF related Soluble factors. And then we, as I showed you in the first data slide, we tried to see if there were any patterns with the ligands being expressed in the tissues as compared to the receptors now on the Tregs. And strikingly, there was quite some alignment. So, and this was significantly different between the ileum and the synovium. So while in the ileum, this was mainly driven by TNF receptor 2 and also trail and light, in the synovium, TNF receptor 2 was really low in the Tregs, confirming our initial results, but also showing that this interaction is more driven by CD30, 41BB, and Treg, also co-stimulatory molecules from the TNF pathway. So that's quite intriguing that these kind of interactions drive the phenotype locally. So finally, we also looked at the signaling pathways. We know TNF signals via the receptors and can induce several canonical pathways like the NF-kappa B pathway. But to make a long story short, what we mostly saw was differences in the MAP kinase pathway, so in the P38 family members. And this is illustrated here, where we also looked at Treg specific responses by comparing the same expression profiles in conventional T cells of TNF ARE mice. And you could see that especially the MAP kinase 11, 12, and to a lesser extent also 14 were more expressed in Tregs from the synovium, whereas MAP kinase 13 is highly expressed in the gut. So there are clearly differences in the signaling molecules being upregulated, potentially by the imprinting of the different inflammatory pathways ongoing in the inflamed tissues. So I hope I convinced you about the following take-home message. So microscopic gut inflammation is clearly important in SPA disease, and we're trying to look into this more and more in other models as well. So the elevated mRNA we discovered on the TNF receptor 2 level was also mirrored by findings we saw in human patients. And in the mouse model, clearly Tregs accumulated, but they had a different functional impact in the joint versus the gut. And this was related to different TNF receptor 2 expression. But also what we saw is the different signaling pathways and the other TNF family member expressed in gut versus joints. So we think these differences can have a major impact on the local immune regulation and could also be the basis to define more specific targets for gut and joint pathology. So by this, I would like to thank you for your attention, and I'm happy to take any questions.
All right, thank you very much.